Hello everyone. And in the previous unit, we covered the network of nerves around your body and divided into different nervous systems. For example, we had three main systems: central nervous system, somatic nervous system, and autonomic nervous system. And in this unit or video, we are going to discover how the nerves are structured and what is the nature of those nerves. And spoiler alert, they are electrical, but I'm going to discover or go through the process of discovery, like how it was discovered that nerves are actually electrical in nature, because we also discussed that nerves carry something, but they do not carry animal spirits or vapors. So what do they carry? And yeah, it was electricity. So was it found out? So here we go. So our scientist is Luigi Galvani. So he was experimenting on frogs, and what in his experiments, what he did was like he gave electrical stimulation to a dissected limb of a frog, and on stimulating that nerve, what we found out was that he observed that there is a movement in a leg because he stimulated the motor nerve. Okay, now the leg didn't twist when the muscle fibers were stimulated. So he found out his reasoning was as follows. Leg moves when nerve is electrically stimulated. Okay. So nerves must conduct electricity. So nerves are electrical in nature. Animals have inbuilt electricity, which is false, but nerves are electrical in nature and it is observable when you kind of stimulate it. And anatomically, we also know that nerves are not similar to muscle fibers. That's why when you stimulate muscle fibers, no response is generated, but when you stimulate motor nerves, a response is generated. Okay, so now we know that the nerves are electrical in nature. We are going to move forward to our next scientist who added on to that, and his name is uh, Hermann von Helmholtz. So, what did he discover? So, his method was he used multiple techniques, mainly to dissection and electrical stimulation. So, what he did was he pinned the frog's corpse on a table and he put electrodes in it at different distances, and he marked all those distances like how far away two electrodes were. And what he observed and what he did was he attached frog's leg with a drum kymograph. Basically, it's a recording device that kind of records when a movement is generated in the leg, and it will mark it out on a paper, a roll. Okay. So what he was interested in it was discovering the speed of uh, nerve conduction, like how fast the nerve conducts the electricity. So he was kind of measuring the time between stimulation of a nerve and calculating speed, like how much time it took a nerve electricity to go across a distance. And this way he could find out what the speed was of nerve conduction. And his reasoning was as follows. So this is the diagram: a frog with a few pins to hold the body, electrodes placed at different distances, and the leg uh, that twitches is connected to a dumb kymograph at the bottom. Okay, so uh, he graphed it out like this. Okay, so whenever there is contraction, it goes up, and then there is a relaxation period. Okay, that is recorded on the kymograph. Okay, so his reasoning was as follows. Okay, on a stimulating nerve, we know there is a muscle contraction. Any form of stimulation, especially in this case, it was electrical, it caused a twitching of the frog's leg or muscle contraction. Okay. The time taken between nerve stimulation and muscle contraction across a distance help us find its speed. Why? Because the speed is distance upon time. Obviously, in the short notes on sub points, I have given how the distance was measured. It was measured between electrodes and nerves uh, on kymograph, and when the paper was kind of spread out like a rectangle, you can actually measure the distance and the time uh, taken. And he had the distance, he had the time through his measuring devices, and then he calculated the speed, and the speed came out to be almost 25 meter per second. So yeah, So we found out that nerves conduct electricity and they conduct it at a specific speed and they are electrical in nature. So now we are going to move forward to our next scientist, and his name is Santiago Roman Cajal. So his method was he took it from Golgi. Basically, you take a nerve cell and you kind of put it under a microscope, a light microscope, and then you observe what kind of formations you see in the cell. And when he took out the nerves and put them under a microscope, he found out there was certain structures, and those structures were made of discrete cells. There was distances in between them; they were defined, they were separate, and those were called neurons. So his reasoning was as follows. We know that nervous system is a network of nerves, and nerves transmit electricity throughout the body, and nerves are made of discrete cells. Let's just call them neurons. Okay. So neurons are basic units that must be involved in the conduction of electricity as well. Why? Because neurons are part of nerves. Nerves conduct electricity, hence neurons also conduct electricity. Okay. The other researcher, which is kind of a sideline point. So basically, Suzanne Hosel, what she did was she took the brain and she kind of converted it into a solution. And then she measured, okay, in a specific volume, how many nervous cells are there, nerve cells. And this way, she could calculate in the entire solution how many cells are there in a average in a brain on average. And she found out there were 86 billion. So through our research by Casal, we can find out, okay, there is a nerve and nerves contain neurons and neurons conduct electricity. Okay. And there are about 86 billion of them in your brain. Okay, cool. And now what we are going to find out is that this is the structure. This is a different form of staining technique, which is called Nissl method, which will you stain it with violet blue, and it kind of helps you observe the more specific parts of the neuron, that is nucleus, cell body, soma, dendrite, etc., axon. Yep. So on observing cells on different neurons on different body parts, in nerves in different places like brain, near muscle fibers, neurons in your spinal cord, we came up with a few different categories of neurons. Sensory neurons in the sensory nerves that carry information to the central nervous system from your sense organs. Okay. Obviously, information goes across a different forms of neurons or a line of neurons, and they are neurons connecting each other. So they are interneurons because they connect one neuron to another. So there is a line of neurons, and these neurons are spread across your body. Okay. And the last one is motor neurons that carry signal from your brain after the information carried from sensory through sensory nerves is processed, and they come out in the form of commands for your muscles to move, and those are carried through brain or spinal cord. Okay. So sensory neurons, they carry sensory information from sense organs to central nervous system. Interneurons are connecting neurons that carry both sensory and motor information, but between neurons, and motor neurons that specifically carry signals from your brain uh, to muscle fibers. Okay, cool. Okay. Now we are going to discuss the structure of a neuron. And the neuron has the following structure. There are axons, there are dendrites, there are cell body, axon, and stuff like that. So what is the reasoning we can do? We know that a nerve is like a wire, neuron conducts electricity, and they carry different form of information. So let's go with our logical deductions. Okay, so we know that nerves transmit information electrically. We verified that through our experiments. Okay. We also found out that nerves are similar to wires, and we know that for a charge to flow, it must be unidirectional. It must go from a positive end to a negative. Okay. So neuron must have a form of potential difference, just like a wire. So there must be potential difference in a neuron. Then we found out that neuron has a start point and an end point for receiving and transmitting information. So we kind of labeled it that dendrites are the ones that actually receive information, and the synaptic, what you call it, synapses are the where the information leaves the nerves. Okay, cool. Okay, information passes through neuron just like a wire. So and we also found that neurons had gaps between them. So information should be able to jump across this gap somehow by some method, which which is that method we will discuss later. But for now, we know that nerves conduct information like electricity in a manner similar to an electrical wire. Okay, cool. So now we can kind of roughly divide neurons into three zones or four zones as you like. Input zone, which kind of receives the information. Integration zone, where that information is kind of capsulated. And conduction zone, where that information goes through the neuron. And output zone, where that information leaves the neuron to some other neuron or some other organ or muscle fiber. Okay, cool. So through microscopes, we also observed there was certain form of cells that surrounded the neurons because neurons uh, kind of function in an integrative manner with different uh, cells as well. So, what type of cells are those that surround neurons? And those cells are glial cells. I'm summarizing what their main functions are. So, epidermal cell basically it is important for cerebrospinal fluid. What does this fluid do? We discussed that it is involved in coolant, works as a coolant, works as a drainage system, as a storage of nutrients, for the brain. Astrocyte, okay, star-shaped, symmetrical, provides nutrients or support. Microglial, they are mainly involved in defensive function or immunity for the neuron. Oligodendral cell, it is asymmetrical but involved in central nervous system, and Schwann cells, they are mainly in peripheral nerves. So yeah, so they provide support to the neurons, and there are two main types of glial cells. One is astrocytes. So what do they do? Okay, we know that glial cells connect neurons to blood vessels. Okay, and these are the type of glial cells that connect a neuron to a blood vessel, and they form a protective layer between neuron and blood cell. So whatever comes through your blood, any form of nutrients or pathogen has to pass through this filter called astrocyte. And this filter helps to protect your neurons from contracting any form of disease or something like that. So they form a blood-brain barrier. Okay, so astrocyte is literally the blood-brain barrier. Okay, between your blood vessels and your brain neurons, astrocytes work as a barrier. Okay, cool. And they can also carry signal from neurons. For example, if neuron is deficient in some form of what you call it, in some form of nutrients, then the signal will go to the astrocyte, which will kind of increase uh, the volume of a blood vessel so more oxygen and glucose can go to the neuron. So that's the main function of astrocytes. Okay, so now we are going to go to our main cells that are uh, oligodendroglia and Schwann cells. Okay, so what do? They form a form of insulation layer across the axon. Okay, so that's what they do. They form a packets. They they surround the axon like packets. They are involved in what you call it, in kind of repairing axon. Suppose a axon breaks, then the glial cells form around the axon or the pathway in which the axon was, and they kind of provide nutrients or any form of material for the axon to regenerate. Okay, so they are repair axon. Okay, and the only two difference is that one is present in central nervous system, other is present in peripheral nervous system. Okay, that is clear. Cool. Now, as you can observe on an overview, we're kind of going in a form of reductionism. We are kind of studying part by part, and we are going on a micro level. We started with what was observable through our eyes or senses, like dissection or anatomy. We were observing the anatomy. Then we went through stuff that was not that easily observable, that was our neurons. And now we are going to go forward on a molecular level. And with each development, it was required that there was new equipments or new form of techniques that were discovered. So, naked eye, light microscope, electron microscope, and so on and so forth. So, now that we have kind of discovered or discussed all those aspects, like how what is neuron, how they conduct electricity, what kind of cells they are surrounded by. Now we move forward to new avenues of research by our new researcher, and whose name is Edgar Adrian. So his main method or discovery was electrical stimulation. He electrically stimulated the nerves of animals, mainly rabbits or frogs, and used amplifiers on oscilloscope. Basically, they are voltmeters or amplifiers are what you call it. Increase the amount of signal. Suppose if there is small signal, it will increase amplitude in nature. So okay, so this is the equipment he used. And what did he observe? He observed that when different intensities of electricity or electrical signals are passed through nerves, the amount of potential uh, difference observed is same regardless of whatever intensity you do. I mean, obviously, if you do too much intensity, the nerve will damage, but in a particular range, no matter what intensity of nerve electrical signal you give to a nerve, it will lead to the same difference or same form of signal. Okay, same potential difference and same form of signal. Okay, the only change was in frequency of signals. For example, the more intense this signal you give to a nerve, the more action potential it produces. Okay, now what he also found out that either the nerve produces a signal or it doesn't at a certain frequency. Okay, so now we go through his reasoning, and his reasoning was as follows: action potential, uh, which is a sudden change in the voltage, uh, occurs when an electrical signal is passed through a nerve. Either it is produced or not produced in a nerve. That is our law, all or none law. And if it is produced, it is same in size regardless of intensity, but the frequency increases when intensity is increased. So that was his findings. Those were his findings, and now we are going to move forward to our two main scientists, that are Huxley and Hodgkin. And what did they discover? Basically, they mainly conducted an experiment on squid eel. They took an axon because axon of a squid is larger than that of humans and more simpler in nature. And he kind of put a micro electrode in it, connected it to an oscilloscope, and this was their method. They did multiple experiments on the axon of a squid, and they put the squid axon in a saline solution or seawater. Okay, micro electrodes were placed at different locations, and a signal was observed. Like, okay, suppose if I give a signal to one side, what kind of action potentially generated on the other side? And they kind of noted all those currents that were generated or all those voltage fluctuations, and then they created a what you call it circuit, and they collected the data and they graphed out like what is the action potential, how far it goes, how low it goes, and stuff like that. So uh, at best, okay, in the most native or primitive stage, what they did was they put one electrode over the axon and one electrode inside the axon, and then they observed what was the resting potential, what was the potential when the nerve was not stimulated, and surprisingly they found out it was negative. Okay, so they found out the membrane potential, that is the potential inside a cell, is negative. Obviously we know that uh, negative potential is due to negative charges. So there must be negative charge inside the cell. So this is the diagram in a very similar manner. Before putting the electrode inside the cell, there was voltage was set as zero, and when the electrode was placed inside the cell, the voltage came out to be minus 60 millivolts. Okay, cool. And this is what they observe. This is the electrode, and this is the axon, and on stimulation action potential occurs. Okay, cool. So the action potential occurs in the following way: This is the basic diagram of action potential. This is the resting potential at minus 70 millivolts. This is the stimulus. This is the field initiations, small current but not enough to generate an action potential, and they noted it. Then threshold. This is the point when uh, this is the point when a signal or electrical signal crosses this point, it causes an action potential even without further stimulation. So they observed it. The process of depolarization. Basically, what happens in depolarization is that the negative potential is reduced and the cell becomes more positive. Okay, so that you can see the potential increasing up to from minus 70 millivolts to plus 40 millivolts, and then the cell again repolarizes, it again becomes negative and it starts to go down, and then it becomes hyperpolarized. It not only becomes negative, it becomes even more negative than it was initially was. It goes below minus 70, and then it again depolarizes, it again start to increase until it uh, attains its resting state, its homeostasis. Okay, so this is the basic diagram. So there are two periods. So basically, in absolute refractory period, what happens is that during depolarization, you cannot trigger uh, any action potential. So during depolarization and repolarization, we call these periods absolutely refractory. No amount of you cannot repolarize, or no amount of stimulus will generate an action potential in the cell during this period. But when it is relatively refractory, which means it is again depolarizing from a very negative state, you can generate an action potential, but it will require huge amounts of stimulus. So these were the findings we found out. Okay, so in the next unit, we are going to cover how what happens inside a neuron, and how does it happen, what kind of ions or anions are used, and how does a neuron communicate across the gap between the neurons to different neurons. Those are we are going to discover. So far we have kind of established that neuron is electrical in nature, because it is a part of nerves, and nerves are electrical in nature, and neurons kind of conduct electricity. They conduct it at a specific speed, and there is a specific action potential that occurs, and there is specific properties of those action potential of neuron. Okay, so this is what we have established so far. Thank you for watching, and see you in the next video.